This is Dr. Pooja, your original guru of biochemistry. Now, first of all, I would like to welcome you all on behalf of DBMCI MDS eGurukul, India's oldest medical and dental PG coaching institute. Now, let's start off with the discussion of the questions. As we all know that first of all, before starting up with the discussion, you people should be clear in your minds that what kind of questions are asked in biochemistry, right? Jैसे questions have been asked on rate limiting enzymes, questions have been asked on oxidative phosphorylation, what are the inhibitors, what are the uncouplers. So these are the most frequently asked questions, right? So let's start up with the discussion. This is first of all grand test 4. Right. So now see, first of all, again, the question is the rate limiting enzyme of lipogenesis. Now, if we talk about lipogenesis, we know genesis is formation. Lipo is fat. So lipogenesis is simple formation of fat. A question is, this is a straightforward question, the rate limiting enzyme. Now, if we talk about this, the rate limiting enzyme, we all know that it is acetyl coenzyme A carboxylase. Why? Because now this is the answer to the question. Now, I would just like to tell you that what are the other important aspects here, what are the other important points that can be asked on this particular lipogenesis. First of all, you people should be familiar with the site because the questions have been asked on the site. कहाँ पे होता है? So this lipogenesis is basically restricted to the cytoplasm. Even though elongation takes place in the endoplasmic reticulum. So if we talk about elongation takes place in the ER. Right? Now, since it is occurring in the cytoplasm, that acetyl coenzyme A, because we know the precursor is acetyl-CoA. This is present where? This is present in the mitochondria and it is impermeable to the mitochondrial membrane. So what happens here is simply citrate shuttle is the one. I'll just write in detail the answer, the explanation to question number 1. So citrate shuttle is required for the transport. Now once this acetyl coenzyme A enters, it undergoes carboxylation in the cytoplasm. So acetyl coenzyme A will undergo carboxylation, which means there is attachment of one molecule of carbon dioxide, right? And this acetyl coenzyme A is converted into malonyl coenzyme A. Now, why this becomes a rate limiting reaction? Because the rest of the function is carried over by the fatty acid synthase complex. Okay, so what are the prerequisites for this carboxylation? We are increasing one carbon atom from 2 to 3, right? So what all we require? We require obviously the enzyme, which is carboxylase. This will be the enzyme, and if we talk about the coenzyme here will be biotin, which is vitamin B7. And always remember, B7 or carboxylation always requires ABC. ABC is the mnemonic to remember. A stands for ATP, B stands for biotin, and C stands for bicarbonate ions. So these three things are required for carboxylation, and this becomes the rate limiting enzyme. Right? So the answer to this question is clear. It is B. Citrate lyase is the enzyme required for splitting of citric acid in citrate shuttle. Malonyl coenzyme A carboxylase is required later, that to decarboxylase, not carboxylase. And CPT1 is for beta oxidation. So lipogenesis ka rate limiting enzyme is acetyl coenzyme A carboxylase. Then moving on to question number 2. This is a very important question from examination point of view, and this has been asked so many times. Biochemical test in thiamine deficiency. Now, if we talk about thiamine, we all know that it is vitamin B1. And we all know that B1 is a coenzyme for oxidative decarboxylation. right? Oxidative decarboxylation, removal of carbon dioxide in the presence of oxygen, and it is required basically for 4 important reactions in the body. But here the question is biochemical test. So if we talk about the biochemical test, it is erythrocyte transketolase test. And this is again a very important question. This becomes an assessment also of B1 deficiency. And if we talk about the reactions, since the question is there, so I would just like to mention that what are the reactions in which it is required. Now, if we talk about oxidative decarboxylation, the first reaction is pyruvate to acetyl coenzyme A. This is reaction number 1, right? Second is alpha ketoglutarate to succinyl coenzyme A. This is reaction 2. Here the enzyme is pyruvate dehydrogenase, which we all know is an example of multi-enzyme complex, where you all have studied TPP is the major coenzyme. Then here alpha ketoglutarate dehydrogenase. Third is transketolase, which is required in HMP shunt. HMP shunt, which plays a very important role in maintaining the integrity of the cell membrane of RBCs. And the fourth one is alpha ketoacid dehydrogenase. Basically, this is required for dehydrogenase for branch chain amino acids. Right? So here if we talk about this, the answer is C option. Right, erythrocyte transketolase activity, which actually tells us the assessment of B1 deficiency. So here the option is, answer is C. Moving on to the next question. Cyanide is an inhibitor of. Before starting up with the discussion, I told you that these are the most frequently asked questions, most repetitively asked questions. Inhibitor of. We all know that cyanide is an inhibitor of, this question is basically from the topic electron transport chain. And in my videos also I have discussed clearly in detail what is the difference between inhibitors and uncouplers. So here the question is simply between what they are asking the inhibitor, and we all know ETC mein how many complexes are there? There are 4 complexes. So if we talk about the inhibitors, I can simply say this is complex 1, right? This is complex 2. We all know that complex 1 is NADH coenzyme Q reductase, complex 2 is succinyl coenzyme Q reductase. They both give their electrons to coenzyme Q, which is ubiquinone. Coenzyme because it is ubiquitous in nature, right? And this coenzyme will then transfer the electrons, because since this is electron transport chain, to complex 3. And this complex will finally hand over the electrons to complex 4. Right? So if we talk about complex 4, simply you can say in the presence of oxygen, metabolic water is produced. And coming over to the inhibitors, if we talk about the inhibitors, complex 1 ka most important inhibitor is rotenone. Second important inhibitor is chloramphenicol. Third important inhibitor is pyridine. If we talk about the inhibitors of complex 2, the inhibitors are carboxin, malonate, and TTFA, thionyl trifluoroacetone. Coming over to the inhibitors of complex 3, all the hypoglycemic drugs, and BAL is there, British anti-leucide, and you can say hypoglycemic like metformin and all. They will be the inhibitors of complex 3. Now the question is complex 4, And a very important question, again so many times asked in the exam, the inhibitors are cyanide, hydrogen sulfide, sodium azide and carbon monoxide. Most of the students get confused between carboxyl and carbon monoxide. You have to understand carboxyl is an inhibitor of complex 2 and carbon monoxide and cyanide are the inhibitors of complex 4. Sometimes a question is asked like this, that complex 4 is basically cytochrome A1 A3 oxidase. This question is also asked. So what is complex 4? Complex 4 is cytochrome A1 A3 oxidase, right? So here you can say the answer will be simply cyanide is an inhibitor of complex 4. Right? Complex 1 is NADH coenzyme Q reductase. Complex 2 is succinate FADH2 reductase. Complex 3 is cytochrome B C1 and complex 4 is cytochrome A1A3 oxidase. So simply the answer here is D. So this is how you have to attempt the question. Clear? Now moving on to question number 4. Again similar to this, basically beta you have to understand, देखो ETC, ETC stands for electron transport chain, right? So whatever energy is involved, I'll just take you here. Whatever energy is given out during the transport of these protons or electrons, this energy is not wasted. This energy is utilized in the synthesis of ATP. So if we talk about synthesis of ATP, that process is oxidative phosphorylation. So how do we define oxidative phosphorylation? Process of formation of ATP. So I can say that ETC plus oxidative phosphorylation is overall known as biological oxidation. First of all, try to understand this fact. Right? So we can say that the most widely used अब question क्या है? Question is asking about the uncoupler. ठीक है? So we can say that if we talk about first of all inhibitor, inhibitor क्या करता है? Inhibitor is blocking a particular complex. So if I talk about inhibitors of ETC, which I have already discussed, okay, so here if I talk about, because you have to be make sure what is the difference. If I talk about inhibitor of ATP synthesis, which is none other than oxidative phosphorylation, it is oligomycin. Right. Now, if I say uncouplers, so first of all, you should be able to differentiate. How do we define uncouplers? Uncouplers are those molecules, the compounds, which uncouple the process. Are those compounds which uncouple the process. Okay, in other words, we can say the energy which was being utilized for ATP synthesis, okay, which was being utilized by ATP synthesis now will be dissipated in the form of heat. Okay, so the energy which was being utilized for ATP synthesis will be now dissipated in the form of heat. And that is why, you know, it is used as a weight reducing drug also. Up coming over to this, uncouplers are of basically two types. Okay, one is physiological uncouplers which are acting in our body. physiological uncouplers. Like, for example, thermogenin is a very important physiological uncoupler. ठीक है, then free fatty acids, right? Now coming over to ये तो हो गए physiological, then there are synthetic uncouplers. Synthetic means which are produced, and the most important synthetic uncoupler is 2,4-DNP, which stands for 2,4-dinitrophenol, right? So there is a difference between uncouplers and inhibitors. Inhibitors are going to block a particular complex, whether it is complex 1, complex 2, 3 or 4 or ATP synthesis. But uncouplers are the ones which are basically uncoupling the two processes. Whatever amount of energy is being released, which was earlier utilized for the synthesis of ATP, in the presence of uncoupler will be uncoupled and it will be dissipated in the form of heat. Now coming over to the options. Options are the most widely used uncoupler of oxidative phosphorylation. Now if we talk about this, just now I told you thermogenin, free fatty acids are the uncouplers, but they are physiological uncouplers. Here the most widely used, so widely used synthetic uncoupler is 2,4-DNP. 2,4-dinitrophenol, and this is a very important and most frequently asked question in the exam. So here we can simply say the answer will be B. Right, one more is there, I'll just mention it. एक atractyloside होता है, atractyloside. This atractyloside is ATP ADP translocase inhibitor. This question also can be asked because this is important. So this is the answer to this question. Then coming over to the next question, coenzyme for transamination. Now this question particularly I have mentioned in my class so many times. A straightforward question is there. First of all, you should be able to know what is transamination. Let me just answer this question. Matlab, you have to make a list of the enzymes along with the coenzymes. So if we talk about transamination, transfer of amino group. So here, matlab, it's like by default. So the coenzyme for transamination is B6. B6 is what? PLP. So the answer is PLP. Now since all other vitamins are also given, I'll just tell you biotin is the coenzyme for carboxylation, because most of the students get confused between this terminology. Riboflavin B2, basically it is used as a coenzyme for electron transport chain, और it's a basically electron donor. B2 जैसे FAD हो गया, FAD will be converted into FADH2, FMN will be converted into FMNH2, like that, right? And coming over to thiamine, the active form of thiamine is TPP, and always remember TPP is required for oxidative phosphorylation, right? And again I just want to mention one more thing. Carboxylation के लिए biotin is required, and for decarboxylation, D माने removal, for decarboxylation coenzyme is again B6PLP, pyridoxal phosphate. This is actually a very important pyridoxal phosphate, a very important coenzyme required for the intake of proteins, right? And for oxidative, there are three terminologies. Oxidative decarboxylation, it is TPP. Clear? And for carboxylation, it is B7. And if we talk about the reactions of transamination, there are two reactions, and transamination is a very important process. ठीक है, transamination, why it is important? There is a transfer of amino group. कहाँ से? From an amino acid, किसको? To a keto acid, to form a new amino acid and a new keto acid. And examples are ALT. We all know ALT stands for alanine transaminase, or the other name of this is SGPT, serum glutamate pyruvate transaminase. And if we talk about the reaction is alanine combines with alpha ketoglutarate. It's a reversible reaction and it forms glutamate plus pyruvate, right? Then second reaction here is AST, which stands for aspartate transaminase, and the other name of aspartate transaminase is SGOT. And if we talk about the reactions here, the reaction is aspartate plus alpha ketoglutarate forms glutamate plus oxaloacetate. Again, since we are discussing these 2 points, you should know one very important thing. ALT is a marker of liver diseases. Marker for liver diseases, and AST is again a marker for heart and liver diseases. And the coenzyme for transamination is PLP. Then transamination के बाद once the transfer is there, then it is removed, and then finally this toxic ammonia which is being circulated from the amino acids, which it will be given out from the body in the form of urea. So urea cycle is basically very important in protein metabolism, obviously. So transamination is one step leading to the formation of urea. So the answer to this question will be simply A, right? The next question is, FIGLU is a test to determine the deficiency of. Again, a very important question, and I have again discussed this in my class so many times. If we talk about FIGLU, first of all, I'll just give you the answer. It is a test to determine the deficiency of vitamin B9. अब ये FIGLU है क्या? What is this FIGLU? It stands for formimino-glutamate. And what is the function of FIGLU? It is basically a test to determine the deficiency of B9. If some subject is deficient of B9, so there will be sure and short excretion of FIGLU in urine. And if we talk about how this FIGLU is produced, so this is produced as a result of metabolism of histidine. Histidine metabolism. So basically there are two questions which can be asked on histidine metabolism. One question is obviously histidine undergoes decarboxylation, means removal of carbon dioxide to form histamine. This is one question which can be asked. And second question, which is there in front of us only, this histidine basically undergoes degradation to form urocanate. Right, this urocanate undergoes simple, it forms an intermediate which is 4-imidazole propionate. When we write something in square brackets, you just have to remember is like that. Now this intermediate forms FIGLU. And this FIGLU in the presence of B9, right, it will give out formimino group. Mimino group. and it will be converted into what? It stands for formimino-glutamate. Formimino nikal gaya, what is left is glutamate. Right, and this glutamate can be converted into alpha ketoglutarate. But here the question is, FIGLU is a test. So if I say B9 is absent, what is the clinical correlation? If B9 is absent, what will happen? The concentration of FIGLU will increase. And if the concentration of FIGLU increases, it is excreted in urine. So I can simply say that excretion of FIGLU in urine is an indication of B9 deficiency. And this test is also known as histidine load test. Very important. Again, this question can also be asked. Histidine load test. Right. So this is the answer to this question. So FIGLU is a test to determine the deficiency of B9. If we talk about, others are also given. If we talk about vitamin A के लिए car and trice reaction is there. I'll just write it here. For vitamin A, car and trice reaction is there. If we talk about B12, for B12, Schilling's test is there. Schilling's test, where isotope used is cobalt 60. And third is the answer itself. And one more is there, vitamin B2. Again a very important question. Vitamin B2 के लिए we carry out this test. Erythrocyte reductase, glutathione reductase test. Glutathione reductase test. Right. So this is also a very important question which is asked in the exam. And since we have written vitamin B2, so I would like to mention vitamin B1 also. For vitamin B1, the test is erythrocyte. Just now we have discussed erythrocyte transketolase test. Again a very important question which you saw that it was asked in this test also. Clear? So here if we talk about the answer is B. Then moving on to the next question. Next question says biotin is a coenzyme of. Just now we have discussed this. If you go to question number 1, the rate limiting enzyme of lipogenesis where carboxylation is there. So biotin is there. Now you see here wherever you will find carboxylase, the answer will be that. Okay? So biotin is a coenzyme for pyruvate carboxylase. Because I told you beta on vitamins, these are the questions which have been asked. And obviously if we talk about if biotin is absent, it will lead to multiple carboxylase deficiency. Because most of the carboxylation reactions are going to suffer in the body. And if we talk about the second enzyme, pyruvate dehydrogenase PDH, which is an example of multi-enzyme complex which requires B1, B2, B3, B5 and lipoic acid. And it requires E1, E2 and E3. PepCK, again an enzyme of gluconeogenesis. And lactate dehydrogenase, an enzyme of anaerobic fermentation where pyruvic acid is converted into lactic acid. So here we can say the answer is A. Always remember biotin is required for carboxylation. right? So here we finish up with the discussion of grand test 4 in biochemistry, and this is how you have to prepare and remember, because the questions are generally asked straightforward in the subject. So I hope this discussion of grand test was fruitful to all of you, and I hope you people are understanding what is this subject like and what are the questions which are asked in the exam and how you have to solve the answers. So wishing you all the best. Thank you so much. Take care.